you know me, I'm always very pleased when we have here someone to talk about viruses. What could be more exciting than viruses, and also more challenging sometimes. Here we have Dr. Ron Geller from Universidad de Valencia, physics, I'm going to introduce it later. Sometimes I wonder how much of what I teach you in the biology course the students will remember like a couple of years later. Anyway, let's just pretend that you do remember something, and in any case, you will enjoy Ron Geller's talk today. Ron Geller is native from Israel, but he got his education and training in California, Santa Barbara and Stanford University. Stanford University at Dr. Judith Friedman. She's a well-renowned researcher. You do protein folding, well, you know, Judith Friedman is a very powerful group in that area. Boris, for some years now, holding a Ramón y Cajal position at the Institute for Integrative Systems Biology. He's the group leader of the biology group. In the last year, he has been working in this amazing world of the viral capsids, that those protein structures that wrap the viral genome. How different mutations can affect the stability, the performance, or the escape of the host immune system or those capsids. Because they analyze not one, but a large number of mutations, they usually need the assistance of computer power. Okay, so, analysis is one of the most interesting topics of research now in biology. So I hope you enjoy most talk, and I will stop talking now on the expert on the world.
Okay. Thanks. So I'm going to give the talk in English. I understand Spanish. So if anybody doesn't understand something, so the idea is that you understand. You can stop me at any point and ask me what the hell am I talking about, and I'll try to explain that. Okay? So to start with, I just want to mention that we're starting a new Master's in Virology program at the University of Valencia this spring. So in case you know anyone that wants to study virology, we have a bunch of excellent virologists that are implicated, and the idea is to give a general understanding of virology, as well as to get practical experience in the lab. Okay? So this is the website, and we have a Twitter account. Okay, so I'm going to talk about RNA viruses, and so I want to just highlight some basic features of RNA viruses that make them unique. Okay, so this is genome size in log scale, and this is the number of mutations, the mutation rate of each of these biological entities. Okay, so here we have multicellular organisms, so very large genomes and very low mutation rates. And here, basically, at the upper left part of the graph, you see RNA viruses. Okay, so they have really small genomes and very high mutation rates. So this means that in general they have very few proteins, 5 sometimes. For example, rabies has 5 proteins, and the things they can do with 5 proteins is amazing. For example, rabies can make a dog salivate, aggressive, and bite in order to spread itself with only 5 proteins. Okay? And they have approximately 1 mutation per genome. Very large population sizes, so 10 to the 10 can be produced in vivo during infection, and very fast replication times. And what this means is that they are the best evolving entities there are. They're excellent at rapid evolution. So this means that they can escape antivirals very fast. As you know, for HIV, you have to give multiple at the same time. Otherwise, you get resistance very, very fast. Okay? They escape vaccine immunity and host immunity, and they're able to emerge into new hosts. So one of the questions we're interested in are how do viruses deal with such high mutation rates? Because basically we know from different studies that in general, mutations are bad. So they reduce the fitness of the protein or the organism. Okay, so this is some work back in the days in VSV, and basically one is wild-type fitness, and they put a bunch of random mutations. You see that most mutations are either lethal or deleterious. Okay? So recently, there are new tools that have been developed that allow you to, instead of putting 20 or 30 mutations, to really ask in a global fashion, type of a systems biology approach, how do proteins affect the function how do mutations affect the function of a protein? And so this has been done in some proteins from HIV or from influenza. And what we wanted to do in the lab, we've been working on picornaviruses forever, and we wanted to address the following questions, which is how do mutations affect picornavirus proteins? How are these viruses able to deal with mutations? Are there differences between different proteins? So the mutation rate is uniform, but are different proteins affected in different ways? And how does the environment where the virus is replicating affect the mutations, the consequences of mutations? So very briefly, picornaviruses are well known. So poliovirus, rhinovirus is the most common human infection, and is a common cold. So these viruses are very simple. Pico, because they're small. RNA, because they have a genome from RNA, and they're viruses, so picornaviruses. Okay, they have a single open reading frame, and that encodes for one protein. And basically, it's divided into three parts, P1, P2, P3. And P1 encodes all the capsid proteins that make the virus shell, whereas P2 and P3, all the accessory proteins. So the way this virus is able to generate all its proteins from one big precursor is because it encodes two proteases that cut the big polyprotein into the individual proteins. Okay? So it's a very compact way of encoding many proteins. So we wanted to focus on the capsid because the viral capsid is really kind of an amazing feat of protein engineering. Okay? So it's very multimeric, so it's 60 copies of 4 different proteins that assemble together, and it's multifunctional. It has to assemble without aggregation, it has to encapsulate the genome, it has to encode the information for binding the receptor, and it has to be able to uncode and release the genome when it reaches the right environment, like the cytoplasm. Okay? So this is a really complicated protein. It's also the key target of antibodies because it's the only protein part that you see in the blood. And it's the basis of vaccines. Okay? And it's, picornaviruses have these icosahedral capsids. So we set out to ask how do these structures, multifunctional, multi-domain structures, deal with mutations. What we did is a technique called deep mutational scanning, which basically involves the following things. You take a library, so you take a plasmid that encodes the virus, and you do some sort of protocol to introduce all possible single amino acid mutations into this library of the virus of interest. You then produce the virus from this plasmid library, and then you have to use a very high fidelity sequencing technique to examine the frequency of mutations. Basically, before selection, so when they're in plasmids, so there's no selection because you don't produce the virus, compared to after selection. So this is conceptually simple. You basically ask at a given position, how many do I see of a mutation in the virus population compared to the wild type. So if normally you have ATG as a wild type, and you ask how many ATTs do I have, okay? So, so you check that after growth through cells or selection compared to the no selection, okay? If the mutation went up in frequency, it's beneficial. And if the mutation went down in frequency, it's detrimental to virus growth. Is that clear? Good. So that tells you that the mutational fitness effect, or MFE, of each mutation. So we did it in the following way. We used a virus called Coxsackie-virus B3. This is a very common pathogen. It's related to polio. It's an enterovirus, and it's associated with severe cardiomyopathy. Okay? So what we did is take the capsid region, and we ordered a set of primers for each position that encode a degenerate 3 nucleotides that are degenerate in a codon matching position. That means that randomly, this primer pool will introduce all possible mutations in that codon. So you pull together 851 primers in the forward direction with a single reverse primer that will generate a bunch of different lengths of PCR products. Each one of these will have a random mutation at the codon that it binds to. Okay? You do the same thing in the reverse direction. This is many plates of primers to do 2 PCRs. Okay? You combine the 4 in the reverse by a PCR with the external primer, and you clone it into a library. Okay? So this way we generated a library that encodes the viral genome with all possible single amino acid mutations. We then produce viral RNA, introduce it into cells. Picornaviruses, the genome is like mRNA, so when it goes in a cell, it gets translated and launches the infection by itself. Okay, so very simple to produce from these libraries the virus. We have to do one more infection in order to link the capsid to the genome, because it could be that you get multiple copies in the same cell, so it could have complementation. And then we get the RNA from here and a PCR of this, and we do the sequencing. And very briefly, what we do, the high fidelity sequencing technique that we do is called Duplex Sequencing. Basically, you get your PCR from the plasmid or the viral RNA, which you do an RT-PCR on, and then you chop it into the right size that you need for Illumina. And then we put unique tags on each DNA molecule, so 12 ends, which allows us, after the PCR amplification and sequencing, to make a consensus. Basically, we can go back bioinformatically and know that all reads that came from the same original DNA molecule. Okay? So you pull them together and you say, okay, all the mutations that aren't part of a consensus, meaning they don't show up in all the reads or most of the reads from the same DNA molecule, just came up afterwards from the PCR amplification or the sequencing. Okay? So we are able to clear away all the background mutations and really keep the real mutations. Okay? There's one problem here with viruses is that the RT enzymes, or reverse transcription transcriptase, makes a lot of errors. Okay? And we start after correcting for errors after the RT. Okay? So we have no way of clearing reverse transcription errors. So we saw that that was a problem. Fortunately, RT never makes 2 mutations in the same codon, because that's very improbable to make 2 mistakes right next to each other. Okay? So what we did was kick out all single amino acid, single mutations within the codon. Okay? So these are the results of that. So okay, so now we only look at 2 mutations per codon, or 3. This is our controls when we sequence the PCR or the RT-PCR. So we have very low mutation rates there. In our 3 libraries, we have a very high, a good rate of mutations. When we sequence the virus population, the wild-type virus doesn't have any mutations. And after selection, we see that the mutation rate went down a lot. This is what you expect. The mutations are deleterious. And so in the 3 libraries, we cover approximately 15,000 different amino acids, and we can track their effect on fitness. Okay? This is 92% of all possible amino acid mutations in the capsid. And as a control, we saw that stop codons disappeared. It was what you expect after growth through cells. Okay? Stop codons are invariably lethal in this virus. So this is the result of a global representation of what we got. So this is the position along the capsid. Each line here is a different amino acid mutation. And this is on a log2 scale. So 0 means it's the same fitness as wild type. The frequency hasn't changed compared to after selection, okay? In the library or after growing through cells. In red, our mutations are neutral or beneficial, and blue is bad. And you see that it's mainly blue, okay? So when we look in the context of normal evolution, so in sequence alignments, so in the red line here is just the average for each capsid position, okay? And in the black is sequence entropy, so how much variation do you see in sequence alignments in nature. And you actually see that they correlate quite well, so these peaks of natural variation in sequences agree very well with the sites where we see these red regions, so they tolerate mutations well. Okay? And which makes sense, they also overlap with antibody epitopes. So where antibodies bind the capsid. These we know are flexible loops on the surface of the capsid that are meant to be able to escape immunity, and so they evolve more quickly. Okay? So then we asked, okay, now we know the fitness effect of many mutations in the capsid. We wanted to understand what features correlate with the mutation being positive or negative. Okay? So we got a very large list of sequence or evolutionary or protein-based estimates. So, and we did some machine learning to try to see what are the most relevant features. Okay? And basically what we found is that with these 5 variables, so natural variation in the viral family in sequences, the amino acid, the wild type amino acid and the mutation, the predicted effect of a mutation on stability, and its surface exposure basically explain really well the effect of a mutation. So we can predict with a random forest model quite accurately, so the Pearson correlation is 0.73, whether a mutation, given these 5 pieces of information, is going to be deleterious or not. Okay? So, after learning this, we wanted to get some insights into some sequence features that are inherent to the capsid. So basically, as I said, that the capsid gets translated. There's a protease that cleaves off the capsid region co-translationally. The first thing that happens is the methionine gets removed and the glycine, which is in position 2, gets myristylated. And we know this is essential for capsid assembly. Okay? And then another viral protease comes and makes 2 cuts. And this takes the capsid precursor and lets it fold into the minimal capsid subunits for assembly. And then you get encapsulation and another cleavage facilitated by the RNA. So we said, okay, can we get some insights into the sequence requirements of these processes in the capsid? So what we did was look at how much each amino acid was enriched in our mutational fitness analysis in the first few sites in the myristoylation. So this is the site that the sequence requirements that ProSite says you need for a myristylation. You have to have the glycine. You can't have any of these amino acids at position 2. You can have whatever you want at 3 and 4. One of these at position 5 and no proline at position 6. And so this was also kind of a validation for us. So the bigger the litter, the more important it is. Okay? And we saw that the glycine was actually the most, any mutation at this position was detrimental, basically. Okay? So you need a glycine at position 1 that makes sense with the ProSite myristylation pattern. And what we saw is that we get very good agreement. So this is S, T, A, G, C, or M can be here, and we see the exact same amino acids. Okay? So we get information about what sequences the virus can tolerate there, and what we see is that position 3 and 4 do actually prefer certain amino acids compared to others. The next thing we did was ask at the cleavage sites what kind of sequences are tolerated that allow the viral protease to cut at those sites. Okay? So this is well known that you have to have a Q and a G in order for the protease to cut. So mutations at these positions are quite deleterious. And we got a lot of information about what's tolerated around the cleavage site. Okay, so with this information, we wanted to ask the following question. We know that the viral proteases have to cut the viral proteins in order for the virus to survive. But we also know that the viral proteases modify the cellular environment to allow for the virus to replicate. So a very clear example is that most picornaviruses don't use cap-dependent translation. And so what the virus does is use the proteases to cut factors that are needed for cap-dependent translation. This basically turns off any translation of those proteins, and then the virus just makes its proteins until the cell explodes. Okay? So we reasoned that since we have all this information about what can be tolerated at each position, we can get that information and search the human proteome for matches. So this is the following process. We generate a position-specific score matrix. All this means is that we take the information from here, and we say, okay, at this position relative to the cut site, we are okay with an L, this a bit less, whatever, and we really don't like these. Okay, so we can actually look for a pattern that we can search with. Okay. So we made a position-specific score matrix. We then used a website called PSSM Search. Fantastic. It queries the whole human proteome for a specific sequence. And we identified a bunch of hits that are predicted to be cleaved by the viral proteases, okay? Including some that were known before. So what we did then was validate some of these. It's not very important, but these are 3 interferon-induced proteins from this list. We look for interferon-induced proteins because those are most likely to be antiviral. Interferon is a very classic antiviral process in the cell, no? So what we show basically is when we express this protein with, so one of the hits was PLSCR1. When we express it by itself, it's this size. When we express it with the protease, it gets cut. And when we block the protease, it reduces the cuts. Okay, confirming that this bioinformatic type of screen can help us discover new factors in the cell that are cut by viral proteases. Okay, and that's what the lead is working on. Okay, so next question we wanted to address is, we know that viruses don't just infect one cell type. Viruses have to transit through the host in order to infect different organs and be able to spread. So the effect of a mutation obviously is dependent on the background. For example, you have a mutation that lets you escape an antibody. So that's very useful when the antibody is there, but when the antibody is not there, that could really be worse for the virus. So we asked what happens when we look at mutational fitness effects in different environments. And we chose these 3 environments. HSP70 inhibition. HSP70 is a chaperone that folds most proteins during their folding cycle have to use HSP70. It's about 1% of cellular protein mass is HSP70. Okay, it's a super abundant, super essential protein. So we use an inhibitor to block HSP70. Okay, to ask what happens when you reduce the cellular capacity for protein folding. We use 32 degrees, that's similar to the upper lungs. Okay, this is a common entry route for viruses. And we use fever conditions as well, which is a very conserved mechanism during infection in mammals. So first we asked, do these environments affect virus replication? And the answer is yes. So this is virus production per mil. 37 is about 10 to the 8, and each of these manipulations has an antiviral effect. Okay, so 70 inhibition reduces almost 2 logs how much virus comes out. Okay, and then we also wanted to ask, do these have to do with the capsid or also other processes? So we took advantage of the fact that the capsid is not needed for replication, so you can actually delete the capsid and put luciferase there. And when you put, this is called a replicon, when you put this into cells, it will replicate because it has all the proteins that are needed for replication, produce a bunch of luciferase, so we can measure whether these different environments affect replication. And the short answer is all of them do. The 32 degrees is the least problematic. So it makes our interpretation of the results a bit harder because this affects the capsid, which is what we are interested in looking at, but also somewhat affect also by replication. But with that in mind, we did the following experiment. We took these libraries that we made before that have all the mutations. We produced the virus now instead of just at 37 degrees, which is what we did before. We produced it in under conditions of HSP70 inhibition at low temperature and at high temperature. Okay. We do one more passage again in order to link the genome to the capsid, and then we do our high fidelity sequencing technique. And these are the results basically. So this is the mutational fitness effect. Again on log 2. So this is wild type at 0. 37 degrees, most mutations are deleterious. When you inhibit HSP70, everything shifts down. So when you reduce cellular protein folding capacity, mutations are more deleterious. When replication occurs at lower temperature, mutational fitness effects shift up, meaning that they're less deleterious. Okay, and this makes sense. Obviously, this is when you have a temperature-sensitive mutant. No, when you reduce the temperature, it's able to maintain its function. No, so this makes sense so that reduced temperature can facilitate the folding of proteins that have mutations. Okay, and 39 was an intermediate phenotype. So this is another way to see the same data, which is basically we look at what happens to mutation in each environment compared to normal conditions. Okay, this is just a bit easier to see. So again, HSP70 is largely worse than under normal conditions. When we block HSP70, 32 degrees are better, and 39 degrees is intermediate. So if you look at the effect versus 37, you have lots of mutations that are now more negative when you block HSP70. Okay? A bunch of neutrals, and very few mutations are better off when you block HSP70. 32 degrees, we have a lot of mutations that now are better, that they have less of a cost. Or neutral, and then 39 degrees was an intermediate effect. And just to tell you that we don't just do computational stuff, we also validate some of these things. So this is just, we take our predicted mutational fitness effect, and we compare it to a competition between two viruses, between the mutant and the wild type virus, to have a validation. So this is the stuff from the deep mutational scanning, and this is a biological experiment where we compete a mutant versus a wild type virus, and we ask, has the frequency gone up or gone down? Okay, this is a fitness assay. And basically, we get good correlations with our predictions. So just a summary of this little part is that the local environment can have an important impact on the cost of mutations to viral fitness. HSP70, here we inhibit it, and we see that everything gets worse. And everything says that this chaperone, what it does is act as a buffer to actually allow mutations to not have such a high fitness cost. So this might be actually a mechanism to allow viruses or mutant proteins to be able to sustain mutations in case they're ever beneficial. Okay? So a sort of a buffer to help evolution. And then the upper lung, so 32 degrees, is actually more permissive to mutations. Okay? Switch gears for a few minutes to talk about immune work that we did with virus, just to explain what we're doing in this slide. So we know a lot about antibodies and how they interact with the capsid. For example, so each of these colors, this is the capsid, is one pentamer, it's called, of the capsid, and each of these regions that's colored has been shown to be, say, a geographical site on the capsid where antibodies bind. Okay? So there are lots of structures of capsids with antibodies, and then we have some data about escape from antibodies. Basically, you take a monoclonal antibody, you neutralize the virus, and then you see what grows, and you sequence it, and you'll get a mutation that escapes the antibodies, always. Okay? So we know a lot about this process, but what we don't really know is what happens in real infections with polyclonal sera. And we also don't know if there are very big differences between species. And we thought that with these very diverse viral populations that have all the mutations in the capsid, we can address these questions. So it's just to show that, like an example of the structure. So this is called the canyon. It's a depression in the capsid structure. Anyway, we have lots of EM data of this. These are the antibodies binding the capsid and where they bind, and so we know that. Okay, so what we did is the following experiment. We take these populations that we generated before. We either neutralize them or mock neutralize them with an antibody or a polyclonal serum. Whatever survives will infect a dish of cells just to amplify our mutants a bit that survive. And then we sequence and we ask, okay, what has gone up here compared to here? Okay, so what has been selected for survival? Okay? And anything that has gone up in frequency after neutralization is because it's resistant to neutralization. And then we can do these type of local plots again where we can see which mutations do this. Okay? So the first thing we did was take a monoclonal antibody from a mouse and do this type of neutralization. So these are different positions along the capsid. And this is the amount of differential selection. So it's positive, so the higher it is, the more it's enriched after we neutralize. Okay? And we see three very clear regions that are enriched in mutations. And there are different viral proteins on the capsid, so that's strange for monoclonal, except that when you look at the structure, you really see that they're in the same epitope. Okay, so these are proteins that come together to form the same epitope. Okay, so the monoclonal behaves as you would expect, and so we see that. And so this is a monoclonal, these are the mutations, and if we put one of these mutations, this S, is a neutralization curve, you really see that it completely escapes neutralization. Okay? So we immunized some mice in the lab to get some antibodies for Coxsackie, and we did this with polyclonal sera for mice. Okay? And what we saw is, so this is the signature of the monoclonal antibody. The mice didn't respond in this region at all, and they all had different mutations at a different site in the capsid. And again, conserved in the same region. And this to me was surprising because this is a virus that replicates in the mice, and they all respond identically. Okay, they all put mutation in a single epitope in the capsid to escape. Okay, and we validated the escape. Okay. So then we wanted to go and ask what's going on in humans. So we got a bunch of sera from the Neutralization. And this is the reciprocal of the neutralization titer. So basically, the higher it is, the better the sera neutralize. Okay. So some don't have neutralization, but many people in Valencia have antibodies against Coxsackie virus. Okay. And for some reason, females tend to have higher titers than men against this virus. Okay. So what we did was take some of these sera. We don't know which virus they saw. We just know that they neutralize Coxsackie. And we did the same experiment, this neutralization experiment. And always humans are more messy. But we see the following. So I don't know if you can see. This is one of the patterns. We see sera that neutralizes really well and has a single epitope that's targeted in the virus. By that I mean that the escape occurs in a single epitope. Okay? We see some that have multiple. So here's another one with a single prominent epitope. Okay? And then we have some that are really, instead of having one large peak, they have multiple peaks in different regions of the capsid. That suggests probably that instead of having a single epitope targeted, they target multiple epitopes at lower efficiency, okay? So this is very recent data that we're finishing. So these map in the same, this one is actually the same as the monoclonal antibody, this epitope, okay? And so now we're really finishing the validation of this to really understand how polyclonal serum target the virus, okay? So that's that, and in the last 1 minute, okay? Where we're going with this, okay? So we did P1, the capsid region, that's 1/3 of the viral genome. We just finished doing the same thing in P2, which is now the non-structural proteins, because we wanted to know, and we're about to start P3. So we want to cover the whole genome to really understand how different viral proteins respond to mutations and how this correlates. You know, so we only analyze the structural proteins, but here you have proteases, polymerases, things that modify cellular trafficking or modulate membranes. So this is where we're going, and this is also very recent data, just to show you. So this is the mutational fitness effect, and these were the structural proteins that we looked at. And in general, they're all negative. Okay, so most mutations are very deleterious. To me, this makes sense because these are really complex structures. Okay, so the idea that they don't accommodate mutations very well makes lots of sense to me. However, when you look at these proteins in sequence alignments, they're the most diverse, which also makes sense because they're the ones that the immune system sees, and so they have to keep mutating. Okay. So now we just got information on these three viral proteins. This is a viral protease, for example, and you see that it's true that different proteins have a different sensitivity to mutations. Okay, so this one accommodates mutations much better. So many are much more neutral. And actually, you see also that it's more variable in nature compared to these other two non-structural proteins to be induced. Okay, so this is where we are with this, and our hope is then to complete the genome and then start to identify with these mutations and then to escape innate immunity. Okay, so we can look for what mutations are enriched when we add interferon or things like this. Okay, so this is the direction we're going in. And that's it. So most mutations are deleterious in some. We kind of do this, but their effect varies by protein, and actually you can predict relatively well what the effect of a mutation will be based on a bunch of structural and evolutionary parameters. Okay. So we see that HSP70 normally should help alleviate the cost of mutations. Because when we block HSP70, everything gets much worse. Okay? That means that normally in cells, this protein is probably really important for buffering or reducing the effect of the mutation, and that's very good for viruses because it lets you maintain mutations that may be advantageous when you confront them with viruses. Okay? Lower temperature is helpful. So mutations are less deleterious when you lower the temperature. Fever-like temperatures don't behave like I want them to, and meaning my expectation was that fever would be detrimental. Okay, so the mutations will have a higher cost when at higher temperatures. And I think this might be because the cells have a heat shock response and upregulate a bunch of chaperones and kind of neutralize the effect of the temperature. And then we see that mice have similar antibody responses, and humans are quite different in how they respond to the immune response. Their antibody response is different. And that's it. This is the lab. Teresa Rodriguez is a postdoc in the lab that did all the neutralization stuff and the P2. And Mutagenesis, our collaborators, which help us with complex things that I don't know how to do, like random forest models and these type of things. And this guy's a physicist. And our funding not drawn to scale, and the Biología for providing us the reagents. And thank you for letting me give a talk, because I haven't given a talk not in front of a computer in 2 years, and I can't pay attention to talks on the screens. So thank you.
Thank you very much. I can't go through a slide that was used in my class. Before we go to questions, I forgot to mention that I read about Ron Geller, by chance, my former student, David Talavera, moved to Valencia to work with Ron. It seems that he didn't have enough viruses with me, so enjoy researching on viruses. One comment before I let it go. It amazes me how you are very setting of plasmids, structures could almost replicate the sequence diversity that is found in nature. But it seems that you got the sequence species that try to connect to sex. Really amazing.
Yeah, actually, you get much more information from this mutational scanning because so viruses have very high mutation rates, like I was saying, but even for viruses, the probability that 2 mutations fall in the same codon, or 3 mutations, sometimes you need 3 mutations to go from one amino acid to the next. The chance that will happen in the same codon is very infrequent because the polymerase will make, okay, the first mutation, the error rate is 10 to the minus 5, so 10 to the minus 4, every 10,000 bases will make 1 mutation. But the chance that the next mutation will be right next to the first one is very low, and the third one is even lower. So you need really 10 to the 11, 10 to the 12 particles or infectious viruses to really sample all the diversity. So we took this data and we used it to ask, does it help inform phylogenetics? So if we take sequences and then we give information that we obtain in the lab, do we get a better fit for how the viruses diverge or whatnot? And we see that this actually informs better, gets a better fit for phylogeny when you give it the information from the lab, because nature doesn't sample everything. Okay.
10 to the minus 4 in 1/3 of around 8,000 of that genome?
The genome is, yeah, 7,000 something.
Okay, we are ready for questions. If you find it difficult to pose your question in English, os dejo que preguntéis en español. There shouldn't be any inconvenience for our guests today, so please go ahead.
Yeah.
Thank you for the talk. Mine is a general question.
Okay.
So, what is happening to the structure of the virus as the mutation is taking place? Do you have the immunity system?
Okay, sorry.
Sorry. What is happening to the structure of the virus as the mutation is taking place? Do you have the immunity techniques you have to visualize the whole process?
Sure. So the question was, what happens to the structure of the virus when we have a mutation, and can we image it? And people can image it. We don't know how to do that stuff. So, but it's possible. The thing is, we have 15,000 mutations, so it's you have to pick the mutation that you're interested in and put a bunch of effort to see one mutation. So it's very hard. What we have is really good structures that are available. So these were some of the first things that were crystallized. You know, these picornavirus capsids make really good structures. Okay, and we have very good computational tools to try to estimate what's going on. But beyond that, we personally don't do any structure. Yeah.
With your virus, what is the ratio between once you infect the cell, how many viruses are viable and how many viruses are not? Because I know for poliovirus, which is probably the same mutation rate, it's 1 to 100.
Yeah. So the question is, what's the particle to infectious particle ratio? And in general, for picornaviruses, it's 1 in 100 is infectious. And I did some work with a different chaperone and inhibiting it, and that goes up a lot. So if you mess with protein folding, the number of non-infectious particles goes up a lot. That said, well, that said, probably the effect is not necessarily only because of the mutation. It's because this is a really complicated structure to fold, and it goes wrong many times. Right? So you can make capsids that actually, so the way you measure the infectious particle to particle ratio is you take the virus, you take your virus culture, you treat it with SDX and with RNAs. So any capsids that aren't well built don't survive the SDS treatment, and the RNAs will eat the genome. And then you concentrate, you spin it down, and then you do a plaque assay compared to taking an OD measurement of the RNA. Okay, so you know how many infectious particles you have, and how many are well-built capsids that have RNA inside, that protect from RNases. And still with this treatment, you have a 1.5. So my feeling is that it's a protein folding problem and not necessarily only the mutation.
But you sequence everything, right?
We sequence everything, yeah. Well, what we have done is to infect cells and then sequence from cells. And that at least tells you that what has gone, like it was able to go in and uncode and replicate, so it was packed in a capsid that was functional. That's what we had done before in these experiments. We take the supernatant and we sequence it.
And then when you're making your recombinant viral particles with your DNA construct, how do you assemble the capsid gene? How do you?
So
is it Gibson? Is it?
Yeah, Gibson is. No, we use hi-fi. So it's seamless cloning.
Then you need to
Yeah.
You're using that?
Marvelous.
Okay, yeah, because that's what I want to use for my stuff.
Do 5 microliter. I hope they're not listening, but you can do 5 microliter reactions instead of 20. It works super well. And yeah, it's very good. And it's very efficient. So we're making very large libraries. You need to not bottleneck at any step. So we buy these really fancy competent cells, and we get 10 to the 5 transformants. So with the hi-fi, there's a big plasmid. If you work with DNA viruses, this is not big, no, but it's 11 kilobases, the plasmid. So we do hi-fi for that, and we get 10 to the 5 colonies from the viruses. Really efficient.
And you do it on one.
We do it on one several times. Yeah.
So you mix all the possible genes, mutations in one reaction with your plasmid, and then
we do PCR. That PCR
the colonies.
Well, we scrape all the colonies into one. While we grow culture, we grow a really grow.
And then you sequence it.
And then we sequence that.
Okay. Cool. Yeah. And my other question was for the human stuff. You said there was a big message.
Yeah.
But these people obviously been in contact with the virus.
Yeah.
But it would have been different viruses, right?
Yeah.
So it's kind of expected what you saw, right?
It's expected.
Because they'll have different polygonal mixtures.
Yeah, the thing is we can only detect things that target our virus. So even if they were infected with another virus, so it could be that that epitope in the other virus was not very immunogenic. That's assuming that the immune system doesn't know how to solve a problem, where instead, when you have one amino acid change, it doesn't know how to target it. I guess it's that it does. So these are things that we're going to do now. So yeah, no. Obviously humans have been exposed, maybe they've been exposed to many things. Actually, we didn't even check yet if these are IgGs or IgMs, which might have some effect. I don't know. But to me it was surprising that the mice respond identical, because this virus replicates a lot in mice. So they all choose the same target. And yeah, in the humans we don't know what they are.
I just thought of 2 things. One was what would happen if you pooled the serum from a whole bunch of humans, and what results you would get. It would be interesting. And then the other thing, how much cross-reactivity will be with any other virus? Because for SARS-CoV-2, what I find in my studies is that a lot of coronaviruses have a lot of overlap.
So we have a bunch of other we have a bunch of other Sakis. I mean, the definition of a serotype many times comes from immune reactivity. So Coxsackie virus B3 in theory is the one that reacts, when we use it, the one that gets neutralized by the serum. On that polyclonal, so we have to see. Basically what we're doing now is we've taken these results, we're mapping the dominance of each epitope in the population. So we're going to test, we're going to make all the mutants, we're going to ask what's the dominant epitope, and how many mutations you need to completely escape neutralization. So what requires for a virus, so we have a polyclonal serum, we know the mutation, so does it just need 1 or 2 to abolish?
Esa es una pregunta, si no lo va a preguntar todo Pablo.
Yes, when you're talking about the temperature, how when the temperature increases, the stop or reduce the mutation, that means that the antibodies are doing this job, even that job is not enough to stop the mutation?
What is doing its job, sorry?
And the antibody.
So we haven't done antibodies and temperatures. So what we saw with temperature, again, this is stuff, so the predicted effect of mutations on proteins largely have to do with protein stability, okay? And so the hypothesis off the bat is you increase temperature, proteins are less stable. If you have a mutation that destabilizes it further, it falls apart, okay? And when you reduce the temperature, the instability gets mitigated because the protein is more compact, no? So that was my hypothesis, and that's like temperature sensitive or cold sensitive mutations. There's a good amount of literature on this with individual mutants. No? So yeah, so what we saw was that higher temperature, I would have expected off the bat to see that everything gets worse. But we don't see that. We see that it's mostly neutral. And my feeling is because when you heat shock, when you put something at a cell at 39 degrees, it doubles or triples its expression. Okay? So it might simply be, and we haven't controlled for that, might be that it just has more checkpoints. So yeah, in terms of the combination of antibodies with temperature, we haven't done that. I don't know if I answered your question. If not, tell me.
Talking about temperatures, I was wondering how to be more sensitive to temperature shift. Cancer proteins, virus, RNA polymers, and viral proteins. All the above.
It's hard
There are many
to.
factors in there.
Yeah, so that's what we were trying to check with the replica. Basically, when you look at virus production, it's more affected by temperature than the replica. But whether it's because you incorporate the capsid with the replication effect or something else, I don't know, so it's harder to tease apart. Picornaviruses in general, except for hepatitis A, which is really, really stable. When you put them at this virus, we did some evolution with these populations to get resistance to temperature. If you put them at 40 degrees for 10 minutes, they're dead. So in general, viruses are really sensitive to temperature, except hepatitis A. Doesn't seem to be. But,
so viral proteins uses chaperones for bonding because when HSP70 inhibits the replication of the virus is affected, right?
yeah.
So that means that they use that system for connect bonding.
Yeah, so that was my PhD. With poliovirus, we showed that HSP70 and HSP90, well, other people show HSP70, and HSP90, which is a down, so you have HSP70 that acts co-translationally and helps most proteins fold, and then there's another really major one called HSP90 that gets proteins from HSP70 to fold, and is also really, really abundant. So capsids for picornaviruses, except for hepatitis A, require interaction with both HSP70 and HSP90. In terms of the polymerase, I think there's also some evidence that the polymerase requires interaction with HSP70. Most things use HSP70. So it's a really central part. It's like ribosome, HSP70, and then do your thing.
Any other question?
Voy a romper el hago la pregunta en castellano.
Sí, sin problema.
Es una pregunta, bueno, primero agradecerte la presentación, ha sido muy interesante. Y es una pregunta general. El viroma saludable que tenemos, los virus RNA que tenemos en el cuerpo, que son saludables, ¿tienen la misma tasa de mutación que los que son infectivos?
Uno, no sé si son saludables, son inocuos. Que hay mucha carga viral neutral, digamos que si tú miras sangre de gente sana, tiene mucho virus, anhelovirus, que es una familia que y no hay ninguna enfermedad asociada con esto, bueno, lo que sepa yo. Entonces, no sé, la tasa de mutación, bueno, tiene diversidad muy alta también, entonces significa probablemente que tienen una tasa de mutación alta, pero no lo sé. porque no se puede cultivar. Entonces se encuentra, son súper comunes, digamos, y gente sabe, en todo el mundo, pero no saben cómo cultivarlos en el momento. Entonces esto hay que medir, porque puede ser que hay mucha diversidad, porque hay mucho, ¿no? Como con SARS, que tiene una tasa de mutación bastante más baja, es un RNA virus, pero es muy grande. Entonces, uno de los pocos, o únicos, que corrige, tienen error correction, ¿no? Entonces, en general, estos tienen muchas mutaciones porque no corrigen errores, les da igual. Y SARS, como tiene un genoma más grande, tiene que tratar de corregir errores, pues tiene menos mutaciones, ¿no? Pero hay tanta infección que al final se ve mucha diversidad. Pero hay variantes que saben, ¿no? Entonces, no te puedo contestar. No sé la respuesta.
Me esperaría mucho que al convivir con el organismo hubiera un equilibrio, ¿no? Y una tasa de mutación no tan elevada, no tendría por qué tener.
Sí. En general son un misterio, ¿no? Entonces, como no son muy abundantes, entonces tienen que replicar en algún sitio, pero no saben dónde replican, no lo pueden replicar fuera. No hay. Y entonces, no sé, la verdad.
¿Alguna pregunta más? Ya que estamos más en Spanish.
Una curiosidad, ¿no? Que siempre nos han dicho, bueno, yo cuando estudié en la carrera, que los virus no son considerados virus. O sea, pero bueno, siempre me han resultado un poco, no sé, porque es que veo que son capaces de hacer tantas cosas, ¿no? Y bueno, quisiera saber tu opinión sobre eso.
Si son vivos o si son.
Sí, porque nos han dicho que para que esté considerado vida, digamos, por lo menos a mí, me han dicho que tienes que tener ambos tipos de ácidos nucleicos y que los virus pues solo tienen solamente uno, y entonces por eso no eran considerados.
Bueno, pero hay virus de ADN que hacen RNA, ¿sabes? Entonces, estos son de RNA, pero hay virus de ADN, entonces, no sé. Yo creo que hay que comer y deshacer de la comida, ¿no? Para servir. Que no sé si estos lo hacen, pero bueno, eso depende de cómo defines qué es vida, entonces, yo de filosofía.
Sí.
Yo en clase digo que no.
Yo, como lo que
Solo unos virus gigantescos que no son normales, amebas, los zoos, esos están en la frontera.
pero tienen que estar dentro de algo para vivir.
Pero
¿No? Porque no vivos mientras están transitando.
No, se lo tiene que hacer una célula.
Entonces,
Una célula proteína.
entonces yo creo que es, bueno, pues, como tú lo quieres definir.
Good question.
Yeah.
Okay, there are no more questions. I just want to thank very much Ron Geller and David for coming all the way from Valencia to here this rainy day. Hope next time will be.